out. So hello students and welcome back again to another video tutorial by Genome Biotech. In our yesterday's lecture, we have talked about what are genes. We have talked about the organelle genome. We have talked about the chloroplast genome. And in today's lecture, we will have to start about genomic mapping. Just to inform you about a error that I had produced in my previous lecture. A gene is always going to start with an exon and it is always going to end in an exon. Okay, so the number of exonic sequences are going to initiate the gene, an exonic sequence is going to terminate it out. That means a gene will start from an exon and ends into an exon. Introns will be present in between them only. Okay, so this would be an exon, this would be also an exon. Sorry for the disturbance caused. And now moving forward, see, we are going to start with genomic mapping. What do you mean by genomic mapping? What are genomic map? See, when you talk about a map, the term map refers to the location of identifiable landmark on a particular chromosome. Okay, so when you talk about map refers to the location of identifiable landmark over a particular area. Any map can predict about the different types of landmark over a particular area. When a map predicts about the different sequences that are present on the chromosome, then the map is commonly referred to as genetic map. A genetic map is the map that predicts about the different sequences that are present over the chromosome, then it is referred to as a genetic map. So a genomic mapping refers to all the different types of maps which are being used to determine the locus of different types of genes in the chromosome. Okay, so genomic mapping refers to the technique which is used to map the entire genome, of an organism, which is used to map the entire chromosome chromosomes. Map the entire chromosome means to determine which are the different sequences which are present in the chromosomes and how these sequences are being arranged. Next, now a gene or a DNA sequence having a known location on a chromosome and associated with a particular trait or gene is used as a landmark. It is referred to as a genetic marker. Now, what is a genetic marker? Genetic marker refers to a gene or a DNA sequence having a known location on a chromosome. When you know about the location of a particular sequence in the chromosome, then that gene can be used as a landmark to make the genetic map. Okay, अगर आपको किसी particular gene का location पता है, then you can use that gene as a landmark to determine the position or the places of other genes. Genes were the first marker to be used to prepare the first genetic map of fruit fly. Mapped features that are not genes but DNA sequences are called as DNA markers. Okay, so genes were the first markers that were used to prepare the genetic map of fruit fly. Mapped features that are not genes but DNA sequences are called as DNA markers. Okay, if there are sequences that are not genes but these are just DNA sequences, then these are referred to as DNA markers. These DNA markers commonly are restriction fragment length polymorphism. When you are going to take the double stranded DNA and you are going to restrict the double stranded DNA with the help of restriction to nucleus, then that is called as RFLP. Simple sequence length polymorphism, also referred to as SSLP. Simple sequence length polymorphism is another type of polymorphism which is present. And single nucleotide polymorphism. The simple sequence length polymorphism refers to as the polymorphisms present in the mini satellite. Mini satellite. Polymorphism present in mini satellite. Polymorphism of the mini satellite. And when you talk about the last one, single nucleotide polymorphism, this is a type of polymorphism that takes place due to a particular nucleotide variation. Single nucleotide variation. Single nucleotide variation is there. Single nucleotide variations occurs. There is a single nucleotide variations that are going to take place. Then it is referred to as single nucleotide polymorphism. Let us have a view about restriction fragment length polymorphism. That what is this restriction fragment length polymorphism and how this restriction fragment length polymorphism is being used. Restriction fragment length polymorphism is actually based upon the number or the different sizes of the DNA fragment that would be produced on the basis of restriction digestion. When you are going to take a DNA molecule, you take a DNA molecule and you now cleave this DNA molecule by restriction in the nucleus. It produces two different fragments, A and B. In another chromosome, DNA has only two of the restriction site. Instead of having three restriction site, there is only two restriction site which are present here. So here you are going to get a single band of DNA present in the form of a C band. This is the C band of the DNA that is being obtained. So this is referred to as RFLP, restriction fragment length polymorphism, which is present. ये क्या है? ये restriction fragment length polymorphism है. There is a restriction fragment which is going to be produced, and because of this restriction fragment, there are several different types of bands that are being produced. Now look here. RFLP arises because the mutations can create or destroy a particular restriction enzyme site. Okay. RFLP arises because mutations can create or destroy the site recognizable by the restriction enzymes, leading to variation between individuals in the length of the restriction fragment. It will produce a variation in the individuals between the length of the restriction fragment produced. A single base change within a restriction endonuclease site will already produce a restriction site or will already reduce a restriction site. Okay, so if there is a single nucleotide change that is going to take place, then there might be a new restriction site produced or there might be a restriction site that could be eliminated. देखिए अगर म्यूटेशन यहाँ पर हो रहा है, if mutation is constantly taking place at a particular site, then there could be a production of a new restriction fragment. There could be a production of a new recognition site by restriction endonuclease. Restriction endonuclease is going to recognize this particular site. RE is going to recognize this site. Then it will cleave this DNA molecule. And here there is no site. Although there was a restriction digestion site present in the parental DNA, this restriction digestion site has been eliminated by the mutation at the site where parental DNA was being cleaved by restriction endonuclease. So such type of variations will be produced when the same genetic material will be cleaved by different types of restriction enzyme. अलग अलग तरीके के fragments produce हो जाएंगे, produced from identical regions of the genome. A single base change within a restriction endonuclease site is a readily detectable genetic marker because of the mutatable sites is no longer cleaved by the enzyme in question. Two chromosomes that differs by such a mutation are then distinguished on the basis of restriction fragment length polymorphism. Two different chromosomes can be separated apart from each other on the basis of the different types of chromosomes which are present. Okay, two chromosomes that differs by such a mutation can be distinguished on the basis of the restriction fragment length polymorphism. On the basis of RFLP, we can separate the two DNA molecules with regard to each other on the basis of number of fragments produced. RFLP has only two different alleles. One, if the site is present, it will produce a restriction digestion and an allele will be present. Okay, a variable allele will be present, such as the allele A and allele B. These are the different fragments which are being produced. Here, these two alleles are absent because there has been a mutation in the site for recognizing the restriction endonuclease. जो साइट रिस्ट्रिक्शन एंडोन्यूक्लियस को रिकॉग्नाइज करी थी उसमें म्यूटेशन हो चुका है and that site is no longer available. Next is referred to as simple sequence length polymorphism. Now, what do you mean by simple sequence? Any query with regard to restriction fragment length polymorphism, you can ask me. Okay. When we are going to use restriction endonucleases against the sites which are being bordered by mini satellite, then the same technique will become DNA fingerprinting. जब आप किसी डीएनए को जिसके अंदर मिनी सैटेलाइट बहुत ज्यादा बार रिपीट हो रहे हैं, उसके एंड्स को आप रिस्ट्रिक्शन एंडोन्यूक्लियस से कट करेंगे, देन द सेम procedure will be applicable in the case of DNA fingerprinting. Okay, then that would be referred to as DNA fingerprinting. Simple sequence length polymorphism. What do you mean by SSLPs? SSLPs refers to simple sequence length polymorphism. Now, simple sequence length polymorphism refers to as the DNA sequence which differs from each other on the basis of their mini satellite. Simple sequence length polymorphism are an array of repeat sequences that display length variations, different alleles containing different number of repeated units. The two different types of simple sequence length polymorphism include the mini satellite as well as the micro satellites. These are the two different types of simple sequence repeat polymorphisms that are present. Mini satellite often used loosely to mean the variable number tandem repeats. These are referred to as the VNTR. And the microsatellites are also referred to as short tandem repeat polymorphism. Microsatellite are referred to as short tandem repeat polymorphism, STRP, or simple tandem repeat, STRs. Okay, so these are referred to as microsatellite, whose repeats are shorter, usually dinucleotide or even tetranucleotide in nature. So their repeats are going to be shorter, dinucleotide or tetranucleotide. Now the next one is referred to as single nucleotide polymorphism. Single nucleotide polymorphism is also referred to as SNP. Okay, this is referred to as single nucleotide polymorphism. Now what is going to happen in the case of single nucleotide polymorphism? There is going to be a mutation. A mutation will be produced at a particular site, at a single site. Then it is referred to as single nucleotide polymorphism, SNP. अगर म्यूटेशन एक पर्टिकुलर साइट पे हो जाए, then it is referred to as single nucleotide polymorphism. These are positioned in a genome where some individuals have one nucleotide, that is guanine. Other individuals have different types of nucleotide, that is cytosine. Then such type of positions contribute to the SNP, single nucleotide polymorphism. For example, you know that beta hemoglobin gene has GAG at its sixth position. If at the place of GAG another sequence GUG is present at the sixth position, then it will be referred to as sickle cell anemia. Then it would be referred to as sickle cell anemia. ये क्या होता है? ये नॉर्मल हीमोग्लोबिन है, and this would be referred to as sickle cell anemic hemoglobin that would be produced at the place of GAG. GUG is being produced. This is the sixth position. So at this particular position, at this particular location, there has been a single nucleotide polymorphism. इस पर्टिकुलर लोकेशन पे क्या हो गया? इस पर्टिकुलर लोकेशन पे हो गया है सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म. At the second position, there has been a polymorphism, and that is referred to as the single nucleotide polymorphism that has taken place. Any doubt, any problem? Next, physical mapping. Now, what is this physical mapping? How does physical mapping take place? And what do you mean by physical mapping? These are all referred to as chromosomal maps. Okay, any doubt? Any doubt? Yes, sir. Drop the message. Okay, no doubt. Anyone else has any doubt? You just ask me. You just can ask me. Now, single nucleotide polymorphisms contribute approximately 1.5% of the total human genome. Total human genome का 1.5% किसका बना होता है? Single nucleotide polymorphism का, which is made up of a single nucleotide that shows polymorphic character. Now, physical mapping. What do you mean by physical mapping? What do you mean by physical maps? A physical map provides information on the linear structure of the DNA molecule. Physical maps are going to provide the information about the linear structure of the DNA molecule. So what is the linear structure of the DNA molecule? How the DNA molecule is being arranged, that type of map which is being made is referred to as a physical map. Physical distance is always being measured in terms of base pair. Physical distance हमेशा base pair के terms में measure की जाती है. Physical mapping uses molecular biology technique to construct a complete genomic map showing the position of sequencing features including genes. Example of most commonly used physical mapping include the restriction mapping, fluorescence in situ hybridization, and sequence tagged sites. Now, there might be a question in your mind. I'm not able to get you, Trishla. Write the message again. Sir, last point is not that you said. What do you mean by last point is not that you said? A physical map provides information on the linear structure of the DNA chromosome. Physical distance is measured in base pair. So the actual map that is being produced is referred to as the physical map. So most SNPs are found in the non-coding regions, such as within introns and in intergenic sequences. So, Trishla, you want to say that, sir, this position which is present is actually present in the axonic regions, and the last, this is present in axon, and it is also present in axon, sixth position of the axon, sixth position of the axon. Last line says that most of the SNPs occurs in the non-coding region. Okay, most of the SNPs, not all SNPs. I was talking about an SNP which is present in the axonic region. मैं एक ऐसे SNP के बारे में बात कर रहा था जो axonic region में है, and this is an SNP which is present in the axonic region, not in the intronic region. Any doubt? Now, physical map measures the distance between the DNA molecule in terms of base pair. It measures distance in terms of base pair. It measures actual distance. It measures actual distance in between gene. Distance in between genes. In between genes in terms of base pair. Of base pair distance. Okay. So the distance is being measured in terms of base pair. This is how the distance is going to be measured. Now, there are different methods: restriction mapping, fluorescent mapping, and sequence tagged site mapping, which is being used. Now, what do you mean by restriction mapping? Restriction mapping uses the restriction enzymes in order to restrict the DNA molecule. Restriction mapping determines the position of the different restriction site in a DNA molecule by analyzing the size of the different restriction fragments which are being produced. It determines the position of restriction site in a DNA molecule by analyzing or by visualizing the different sizes of the restriction fragments which are being produced. You look at hint third. This is a enzyme that is going to cleave the DNA molecule. The DNA molecule will be cleaved in two different fragments, one of 2.8 kilobase pair and another one of 1.2 kilobase pair. These are the two different fragments which have been produced. Now, when you use a combination of hint third and BAM H1, जब आप hint third और BAM H1 का combination use करते हैं, then you will get the following different types of bands: 1.8 kilobase pair, 1 kilobase pair, 0.9 kilobase pair, 0.3 kilobase pair. These are the different bands which have been obtained. Okay, then when you are going to use BAM H1, you obtain 1.8, 1.3, and 0.9 kilobase per band. Now, just try to reconstruct this map. Just how you are going to reconstruct this map. Please look here. Look here. See this. This would be the cleavage site of hint third. This would be 2.8 kilobase per, 2.8 kbp. And another would be referred to as 1.2 kbp. The next one, BAM H1, is referred to as 1.8, 1.3, and 0.9. 1.8, 1.3, and 0.9. And BAM H1 and HIND third. When you are going to cleave the BAM H1 when HIND third, then how many number of fragments will be produced? See, 1.8 base pair, 1.8 kilo base pair fragment is intact. देखो ये वाला fragment तो intact ही रहा, क्योंकि ये वाला fragment intact रहेगा. When this fragment remains intact, I am just labeling those two fragments. I have already completed this one. I am just comparing both of these. So when Which fragment is going to remain constant? Tell, look here. It is this fragment, 1.8 kilobase pair, 1.8 kilobase pair. That is going to remain same, 1.8 and 1.8. It is going to remain same. These are the two DNA molecules. Now, one 0.9 and 3 base pair. Okay. Now, when you talk about 0.9, 1, and zero point 0.3, then how it could be produced? The same could be produced when this 0.91 would be broken down. How? See, this would be of 1 kilo base pair, 1 kbp. This would be of 0.9 kbp, BAM H1 and Hind third. BAM H1 and Hind third are being combined. Na? Okay, so this is 2.8 ATP. 2.8 kilobase pair would have been produced. Okay, but out of this 2.8, when 1.8 has been subtracted, देखिए क्योंकि इस वाले के अंदर, इस वाले ग्राफ के अंदर देखिए what are being mixed, which are the two different restriction enzymes which have been mixed. BAM H1 and Hind third have been mixed. That means BAM H1 and Hind third has been mixed. Sorry, these are the two which have been mixed. So firstly, the DNA is going to be cleaved with Hind third. I'm just erasing all other things. Okay, firstly, the DNA is going to be cleaved with Hind third. Hind third will produce the following bands: 2.8 kbps band, that would be of this particular side, and 1.2 kilobase pair size band. Okay, out of this 2.8 kilobase pair band, there would be two different bands that would be produced. Okay, this and this have been combined together. Sorry for the disturbance caused. These two have been combined together, mixed together, mixed together, and then used. Then what is going to happen? 2.8 and 1.2 is going to be cleaved. Now this 2.8 would be cleaved in the form of 1.8. Okay, it would be cleaved in the form of 1.8. The rest one was 1.2. Now, how this 1.2 is going to be cleaved? The 1.2 would be cleaved, 1.2 can be made to cleave into 0.9. Okay. So this should form 0.9, and this would be forming 1.3. But this 1.3 will also be cleaved because of the 1.2 kilo base pair region. 1.2 की वजह से ये भी cleave हो जाएगा. And there are how many number of cuts produced? 1, 2, 3, 4, 5. 5 cleavage segments will be produced. 1, 2, 3, 1, 2, 3, 4, 5. These are the 5 cleavage segments produced. Because of these 5 cuts that are going to be made, क्योंकि 5 कट होने वाले हैं तो 5 fragments produce होने वाले हैं. You can find this BAM H1 fragment 1.8 being made here. There is another fragment 1 which has been made. 1 is this particular fragment, 0.3 is this particular fragment, 0.9 is this particular fragment, this fragment is 1.8. Okay, then you have the other fragments which are present. 1.2, 9 plus 3 equals to 1.2 kilo base pair fragment, and these are the other fragments, 0.9 base pair fragment. So that would be the construction of the restriction map. If you feel any difficulty in drawing this map ना, अगर आपको लगता है कि आप पे एग्जाम में होगा नहीं होगा कैसे होगा, तो मैं आपको एक ट्रिक बताता हूँ. आप इसको पहले ऑप्शन में चेक कर लीजिए, फिर ऑप्शन को आप रिवर्स अप्रोच से क्वेश्चन के साथ में मिलाइए. यू जस्ट check the all possible options that are present and then match the options available with the question restriction digestion sites. Then you will be able to predict whether the digestion of the DNA is possible or whether the digestion of the DNA is not possible. क्या डीएनए का डाइजेशन होगा भी या डीएनए का डाइजेशन नहीं होगा? It all depends upon the restriction digestion system. Okay. Any doubt, any problem? You just look at the option, then match the option with the question. and you would be able to get the correct answer. Okay, this has not been made clear. This is clear to you. This is clear to you. Okay, now what we have to make is we have to make restriction digestions. Okay, now how restriction digestion has to be made? Okay, so when you make the restriction digestion with Hind third and BAM H1, Hind third produces a fragment which is equivalent to 2.8, and another fragment which is being produced is that of 1.2. See this. This is the fragment of the Hind third. पहला फ्रेगमेंट हिंड थर्ड ने यहां पर बनाया, दूसरा फ्रेगमेंट हिंड थर्ड ने यहां पर बनाया. This fragment would be of 1.2 from here till here. This fragment will be of 2.8. Is this clear? Okay, probably this is clear to everyone because of hint third. Now, I'm taking another color, that is a red color for the BAM H1. Okay, for the BAM H1, I'm taking a red color. Now, where does this BAM H1 is going to cleave? The BAM H1 is going to cleave at a distance of 1.8. Okay, so this is the region where BAM H1 has cleaved. This is the region where BAM H1 has cleaved at 1.8. 1.8. After 1.8, where the BAM H1 is again going to cleave? BAM H1 is going to cleave at 1.3. Okay, you just look here. If you subtract 2.8 from 1.8, what you are going to get is 1. Okay, this is the size that you are going to get, but the other cleavage is going to be made at 1.3. So 1.3 means here, this is the cleavage site. This is the actual cleavage site which is being generated due to BAM H1, and this is the cleavage which has occurred when BAM H1 has made the cleavage. Then 1.8, then 1.3. Where is 1.3 lying? 1.3 cleavage is going to take place here. Okay, here is the cleavage going to take place, but this cleavage is a cleavage which is produced as a result of Hind third. ये क्लीवेज किसकी वजह से हुआ है? हिंड थर्ड की वजह से हुआ है. देखो ये रहा ये क्लीवेज, ये रहा ये क्लीवेज. कितने पे था? 1.8 and 1 was equivalent to 2.8. Okay, so this is the region that we are going to talk about. अगर अभी भी दिक्कत है तो मैं इसे हटा देता हूँ. देखो ये रहा. This is the region that has been cleaved. Okay, 1-to-1 relation. 1.8 cleaved by again another one to give 2.8. Here is the cleavage region. What remains from here is 1.3. So this remains to be 0.3. Okay, and because this region was 1.2 kilobase per long and 0.3 has been separated, so what remains here is 0.9. Any problem, Trishla ji, now? Any problem, Trishla ji, now? Hello, hello, do you have any query? No problem. Okay, is it clear now? Is it clear now to everyone or not? क्या ये सबको क्लियर है या नहीं है? Should I move forward? Okay, moving forward. Now look at the next question which is given here. I've already told you that this one was the question that you had to solve. Now this is the next question that was given to you as a homework. MCQ booklet, chord curve questions, test and evaluation, transposons, genes, genome, and mapping. MCQs were being told that you must solve them all. Now the next point, a purified DNA molecule. Okay, I'm just solving this particular question. If I could trying to solve this one. A purified DNA is subjected to restriction endonuclease digestion by various times, which generate partially to fully cleaved DNA molecule. So there are several different types of DNA cleaved DNA molecules that have been cleaved. Which are the different DNA molecules that have been cleaved? You have taken a purified DNA molecule and cleaved it with several different types of restriction nucleus. Okay. The fragments are separated by agarose gel electrophoresis and stained with Etherium bromide. The fragments which are being produced, they are being separated by agarose gel electrophoresis and then they are being stained with ETBR. The bands are visualized under a UV light source and photograph. Find out the total number of bands which are being produced in the photograph. कितने अलग अलग तरीके के bands produce होंगे. Okay, you look here at the partial digestion. You look here at the complete digestion. The complete digestion will produce the following different types of bands: 1.2, 2.6, 2.2, 4.1. So how many different bands? 1, 2, 3, and 4. Four different bands are going to be produced. 1, 2, 3, and 4. So these are the four different bands which are going to be produced. ये वो चार बैंड है जो कि प्रोड्यूस होते हैं. Which are these bands? 1.2 kilobase per band, 2.2 kilobase per band, 2.6 kilobase per band, and 4.1 kilobase per band. Now, what about partial digestion? In the case of partial digestion, sometimes the digestion will take place and sometimes the digestion is not going to take place. Now, which are the different sequences that can be introduced in the case of, that can be introduced in the case of partial digestion? Now, supposingly for calculating partial digestion, you just have to name them A, B, C, D. A, B, C, D. Now make the different types of combination. Now, supposingly that cleavage occurs at A and B site. Okay, you just have to name them A, B, C, D, and then where does the cleavage is going to take place? If the cleavage is going to take place at A site, B site, C site, or at D site. So how can the cleavage takes place and where will the cleavage take place? Different combinations have to be made and the different combinations have to be solved. Okay, so if the cleavage is going to take place at these particular DNA sequences, B is going to be combined with D. This is the B DNA. Okay, so they have named it differently. I'm just using their name, which Pranav sir has named, so that it will become easier. So Pranav sir has named all these DNA molecule in the following order. He has named the smallest being 1.2. I'm also labeling it like that only. Smallest being 1.2. This is A. The second being 2.2, that is B. The third being 2.6, that is C. And the fourth being 4.1, that is D. Now, which are the different combinations? A. A is going to combine with C or D. So A can combine with either C or A can combine with either D. Okay, so see this. A can combine with C, A can combine with D. D can combine with B, D can combine with A. Okay, so D has been combined with A. Now D can combine with B. So first this is being done. Okay, I have made this combination. Its combinations have been made. A has combined with C, A has combined with D. Okay, now moving forward, D has combined with A and D has combined with B. So because D has already combined with A, so we are going to combine D with B. D is going to combine with B. B and D are going to be together with each other. Okay. Now B can either combine with D or B can either combine with C. So B has already combined with D. So B is going to combine with C. B अब किसके साथ में combine हो जाएगा? C के साथ में. Because C has been combined. C can either combine with A or C can either combine with B. C has even combined with A and C has even combined with B. So these are the four possible doublets that could be made. Now, what about the triplets? Now, how the three triplets can be made? Okay. A can combine with A, D, and B. Okay. So it could be A, D, and B. A, D, and B combining together. Okay. So that would be A, D plus B. A, B, and D. A, D, and B. Or it could be B, C, A. That means A, B, C. A, B, C combining together. Or it could be A, B, and C combining together. Or it could be C, B, D combining together. C, B, D, C plus B plus D that could combine together. Now, these are the different combinations that could be produced. So these are the different combinations that have been produced. CBD has been produced. ABC has been produced. Can you make ADB? ADB. ADB. Okay, we can make that ADB as well as, or it could be AC plus D. A plus C plus D. Okay, this could be another combination that could be produced. A, C plus D. These are the combinations that are going to be produced in the case of partial digestion if going to take place. Okay, A, C, and D. How? A, C, and D are going to be joined together. Here, the cleavage is going to take place. And A, D, B can take place if here and here the cleavage is going to take place. So there would be several different types of fragments that would be produced. You can now just add on their size and the fragments will be produced. So instead of all these things, there is another fragment which is being produced that is referred to as ADB. A, D, A, D, and B. Okay. ADB. A, D, and B. This fragment will also be produced. These are the 8 different fragments. 1, 2, 3, 4, 5, 6, 7, 8. 8 different fragments would be produced. And ADB will be equal to A is A, D, and B. 7.2245. These are the different fragments that would be produced. And this would be the genomic DNA map that has been produced. Any doubt that you have? ये देखिए अलग अलग फ्रेगमेंट्स कैसे प्रोड्यूस होंगे, ये बस बनाया गया है, यहाँ पर कॉम्बिनेशंस बनाए गए हैं, इन कॉम्बिनेशंस के बेसिस पे बोल दिया है कि कौन कौन से फ्रेगमेंट्स प्रोड्यूस होंगे, which are the different fragments which are going to be produced on the basis of these combinations we would be able to calculate. In this diagram, sir has not been able to form another fragment that could be produced, that would be ADB. This is the fragment which is being produced. I have produced one more fragment that is referred to as ADB, and this fragment will be A plus D plus B, that is 7.5. This would be another fragment that would be produced. Any doubt? Any doubt? Any problem? No doubt now. Is this clear to you or not? Is this clear or not? Okay. Now moving forward. What do you mean by fluorescence in situ hybridization? Fluorescence in situ hybridization, as you know, is commonly referred to as fish technique. Now, what happens in the case of fluorescent in situ hybridization? In situ hybridization means within the cell, the hybridization is going to take place between the genomic DNA and the fluorescence dyes. So, fluorescence dye is going to stain the genomic DNA. Then the technique which is being produced is referred to as fish, also referred to as fluorescence in situ hybridization technique. इस टेक्निक को हम क्या कहते हैं? फ्लोरोसेंस इन सीटू हाइब्रिडाइजेशन. In this, the fluorescence tags will be there, and this fluorescent tag is going to be marked at a particular region in the chromosomes, and these fluorescence labeled DNA probes will mark this particular tag. It involves hybridizing a fluorescence labeled DNA probe to denatured chromosomal DNA at the metaphasic chromosome. Okay, at the metaphasic chromosomes, the DNA probes are going to be bounded to the metaphasic chromosome. In the past, probes were labeled with radioactivity. In the past sequence, probes were there and probes were labeled with radioactivity. But nowadays, fluorescence dyes are used for increasing sensitivity as well as resolution. In fluorescence in situ hybridization, the DNA probes is either labeled directly by incorporating a fluorescent labeled nucleotide precursor molecules or by incorporating a nucleotide precursor molecule. Okay. The portion at which probe hybridized to the chromosomal DNA is visualized by detecting the fluorescence signal emitted by the labeled probe. The probes can be effectively identified by detecting the signals which are being produced by the probe. Fluorescence in situ hybridization enables the position of a marker on a chromosome or extended DNA molecules to be directly visualized. So, it is a technique that can be used to visualize a particular DNA molecule within a array of chromosomes. So, within the chromosomes, you can visualize the DNA molecule through fluorescence in situ hybridization technique. Okay. Now, there is another type of tags which are being used. One is referred to as sequence tagged sites. Now, what do you mean by sequence tagged sites, which are also referred to as expressed sequence tags? So, in this fish, how the chromosome is going to appear? Sorry, improper diagram made. Just look at the diagram. This is a chromosome. When you look at a chromosome, then the tags, probe tags, will bind to the chromosome at different locations. There would be different probes that would be used, and these probes, sorry, sorry, in these probes, these different types of probes are being used. These probes will mark the chromosomal DNA at different location. Any of the DNA molecule can be effectively marked and identified using these probes. Okay, so the DNA molecule can be effectively marked, it could be identified using the different types of probes. Now, what do you mean by sequence tagged site mapping? The sequence tagged site mapping, also referred to as the STS mapping. The sequence tagged site mapping, also referred to as STS mapping, tracks the genomic DNA that is operationally unique and has a single occurrence in the sequence of the genome. So there are certain sites which only occur for once in our entire genome, and those sites are referred to as EST sequences, also referred to as expressed sequence tags. STS are detectable by polymerase chain reaction, are useful for localizing and orienting the mapping and sequencing of the data. They are being used in localizing as well as orienting of the mapping. How can you localize the different genes? How can you orient the different genes for mapping? As well as how can you sequence the entire database? Then they are being used in all these purposes and are referred to as sequence tag site mapping DNA molecules. Okay, STS molecules. They occur only uniquely within the genomic DNA, and they are going to be expressing a particular protein molecules. Next, a map representing the order and spacing of sequence stacked sites within a stretch of DNA is known as STF mapping. If you're going to construct a particular map that will depict the different stretches of the DNA that has these STS sequences, then the map is referred to as STS mapping. Okay. Now, there is another type of map which is being made, and that is referred to as genetic mapping. Now, what are these genetic maps? How genetic maps are being made? Genetic maps are the first genetic maps were being made by Strutiven. These were the first maps in order to locate. These were the first maps. These were the first maps made made in order to determine determine the distance in between the genes, the distance in between the genes, in between the genes, on the basis of in between the genes, on the basis of distance in between the genes is being calculated on the basis of frequency of crossing over. of crossing over in between them. So frequency of crossing over can be calculated, and then it could be used in the form of a genetic mapping. Next, organisms contain far more genes than chromosomes. These are all part of the alleles and can assort independently. Human genome has approximately 23 pair of chromosomes and approximately 35,000 number of genes are present. Okay, more than 50% of the of more less than 1% of our genome is coding, and more than 98% of our genome is non-coding in nature. Genes that are closely related on the same chromosomes stays together. Okay, so those genes which are going to be closely related, so supposingly there is a gene X and this gene is closely related, this is closely related genes, closely related genes, these genes are closely related, these are closely related genes which are present. Okay, then these genes are said to be closely related genes, they will stay together. Whereas the genes C and D that you can find here are quite farther apart from each other. Because the genes C and D are quite farther apart from each other, so these C and D genes will undergo crossing over. Crossing over between locations on homologous chromosomes is going to take place. So crossing over is going to take place between the homologous chromosomes. Okay. So 23 pair of chromosomes contains 35,000 number of genes. All the genes which are present over a single chromosome constitute a single linkage map. The linear order of genes along the length of a chromosome can be constructed. Ordering of genus along a chromosome is called as the genetic map, also referred to as linkage map. If you are going to determine the order of genes along a map, then such type of map is referred to as a linkage map. अगर आप किसी भी सीक्वेंस का जीन्स का सीक्वेंस बता रहे हैं एक पर्टिकुलर क्रोमोसोम्स के ऊपर, all the genes which are present over a particular chromosome constitute a single linkage group. I've already explained this in the class of genetics, that in the case of males there are 24 linkage groups, because in the case of males there are 22 autosomes and there is an X and there is a Y chromosome, whereas in the case of this is 24 linkage groups. Whereas in the case of females, when you talk about the females, females contain 22 autosomes plus XX. So they contain 23 linkage groups. 23 linkage groups. These are the linkage groups which are present. And these are 23 linkage groups which are present. Okay. Linkage map is expressed in terms of map unit. Map unit is being expressed. Map unit, also referred to as centimorgan, is the genetic distance, an average of one chromatid of 100, one chromatid out of 100 will have undergone a crossing over of event. That means, see, 100 में से एक chromatid के अंदर crossing over event होगा, then the map unit is said to be one map unit. If out of 100 base pair distance there is a frequency of 1% crossing over, then the map unit distance is being calculated to be 1 centimorgan. 1 centimorgan means the probability of occurrence of crossing over is just 1% in between the two DNA sequences. Okay. Now, it was Alfred H. Sturtevant who constructed such type of chromosomal map. Such type of chromosomes map were being constructed by Sturtevant under the guidance of Thomas H. Morgan at the laboratory at Columbia University. Okay, in 1911, he prepared the first genetic map of the X chromosome of Drosophila melanogaster. So, the first genetic map was that of the X chromosome of the Drosophila melanogaster. So, this is how genetic maps were being constructed. So, these are the different types of genomic sequences, and this is the genetic map that is being constructed. Here it is through these genetic map and genomic sequences that is why we can identify which are the genes which are present over the chromosomes and how those genes are being aligned over the chromosome. Chromosomes के ऊपर कौन कौन से genes होते हैं? What is the locus of genes? What is the locus of genes? What is the exact locus of genes? Locus of genes. How that locus is organized? How locus is organized? Is organized, that we have to determine, and that would be used to determine the position, the locus, the identification of different type of genomic sequences, as well as calculating the crossing over frequency in between them. Calculating crossing over frequency. This is all that could be effectively determined. Any doubt, any problem, you can just ask me. The next topic is about DNA replication. We are going to start DNA replication tomorrow only, because I will finish this entire topic of DNA replication. Not entire, but at least the enzymes of DNA replication tomorrow, and then we'll move forward. Okay. Any doubt till now? Any doubt till now? I knew that this topic was not so interesting. That is why you might have faced some problems. If you still face some problem, you just ask me. Okay, so I'm going to end here only, and we will start this DNA replication tomorrow with certain questions. So I would be sending you some questions about this particular process. Okay, so we must start. Okay, we are going to start here only. So DNA replication. What do you mean by the process of DNA replication? DNA replication refers to a process which is involved in the replication of the genomics DNA sequences. Transmission of the chromosomal DNA from one generation to another is very important phenomenon, because without the transfer of the genetic material, the cells cannot replicate. Okay, so thus when chromosomal DNA is accurately replicated, then only it will provide two cells the entire genome for the faithful distribution into each daughter cells. Only when the genomic DNA has been replicated, then only the two daughter cells will be produced. Else the two daughter cells can never be produced. So for explaining the DNA replication process, there were several different types of models that were being proposed. The first model that was being proposed to explain the process of DNA replication was referred to as the disruptive, the conservative, and lastly, the semi conservative model of DNA replication. Now, what are these conservative and dispersive process of DNA replication? In the conservative process of DNA replication, firstly, let me talk about dispersive. According to the dispersive process of DNA replication, there would have been a cell, there would have been the genetic material present within a cell, and this genetic material would have been fragmented into two. Okay, one and another. It has been broken down into two. This broken down genetic material would have been inherited by two different cells, one here and another here. Later on, this broken DNA will grow and will become a full size. This DNA will also become a full size, producing two different daughter cells. But this method is entirely wrong. The information which is present in a DNA molecule cannot be distributed just by breaking it into two different parts. You cannot just break the DNA molecule into two different parts. So this method has been outdated. It has not been considered that this is how the process of DNA replication would have taken place. Then there was another method which was being known as conservative model of DNA replication. According to the conservative model of DNA replication, it was being thought this is dispersive. According to the conservative model, according to the conservative model, according to the conservative model of DNA replication, there is the double stranded DNA which is present. If this is the double stranded DNA which is present, this double stranded DNA will divide. Okay. When this double stranded DNA is going to divide, one strand will be inherited by one daughter, another strand will be inherited by another daughter strand, another daughter cells, and then the complementary strands will be biosynthesized. इसके आगे इनके complementary strands biosynthesized कर दिए जाएंगे. Their complementary strands will be biosynthesized in such a manner that this is the strand of DNA molecule. Over it, another DNA molecule strand will be biosynthesized. This is the strand of DNA molecule over which another DNA molecule strand will be biosynthesized. This is one DNA molecule. This is another DNA molecule. Any doubt? But such type of method of DNA replication cannot be true because you cannot separate the two strands away from each other just like that. And then the complementary strands cannot be biosynthesized over them. ऐसे ही आप डीएनए को सेपरेट नहीं कर सकते और ऐसे ही इसके ऊपर एक कॉम्प्लीमेंट्री स्ट्रैंड नहीं बना सकते because this process is very energy expensive, highly energy expensive, highly energy expensive, expensive, so cannot be used, so cannot be true. So this cannot be true. Okay, the next method that was being explained by Watson and Crick and ultimately being proved by Michelson and Stoll is referred to as semi-conservative model of DNA replication. According to the method that is referred to as semi-conservative model of DNA replication, the two strands of a parallel double helix DNA molecules have anti-parallel nature. The bases on one strand are complementary to the bases on the other strand. The basis sequence of each parental strand could serve as a template for the synthesis of new complementary strand, producing two identical double-stranded progenies. This process is called a semi-conservative model of DNA replication. Each parental single-stranded DNA is going to remain intact. The alternative method which are being used are referred to as conservative and dispersive mechanism of DNA replication that I have already explained. What happens in conservative replication? The whole original DNA sequence is being remained intact. Conservative replication in which the whole genomic original double helical DNA act as a template for new synthesis. Okay. In dispersive replication, some part of the original DNA helix are conserved and some part are not. It was Michelson and Stoll who experimentally proved that the semi-conservative mechanism of DNA replication holds true in the case of E. coli. किसके अंदर ट्रू होता है? E. coli के अंदर ट्रू होता है. Which is the growth medium that is being used? The growth medium that is being used is referred to as the ammonium chloride. This is the ammonium which is being used. NH4Cl. So what would be the ammonium chloride molecule? This would be the ammonium chloride molecule. NH4Cl. This is the ammonium chloride that is going to be used. Now, when this ammonium chloride is going to be used, you have a double stranded DNA molecule containing nitrogen 15, heavy nitrogen N15, and another one is also referred to as N15. This is N15, this is N15. Now you have to grow it into ammonium chloride medium, that is N14 medium, NH4Cl, and this NH4Cl is N14. Another one is N14, NH4Cl. Okay. Now this is NH4Cl and this is NH4Cl. In both the medium, what is going to happen? One strand will remain as the parental strand and the other strand will be biosynthesized. एक strand तो parental रहेगा ही रहेगा और इसके सामने दूसरा strand क्या हो जाएगा? The other strand is going to be biosynthesized. This is the other strand that has been biosynthesized. Parental DNA and the other DNA has been biosynthesized. So this is the double-stranded DNA molecule which has been produced. So this would be referred to as N15, N15 type. This would be referred to as N14, N15 type, N14 and N15 type. N15, N14 type. And the last one, when again it is going to be grown in ammonium chloride, NH4Cl, and that too ammonium chloride, that is 14, it will remain N14, N14 type, N14, N15 type, N14, N15, and N14, N14. So what you get concluded from this particular diagram is that only one strand of the parental DNA is being copied, and another strand of the parental DNA is and another strand of the daughter DNA is being produced. एक strand को copy कर लिया जाता है and the new strand is biosynthesized. One strand of the parental DNA goes to one daughter cell, another strand of the parental DNA is being inherited into another daughter cells. The complementary strands are being biosynthesized, and that is why you have a double-stranded DNA molecule, of which one is the original, another one is the newly produced DNA molecule. Okay, so here you have taken N15, N15 type, you got N15, N14, N15, N14. In the other case, the next cycle, you again took N14 type, you get N15, N14, 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 N15, N14, and N14, N14. So these are the different double-stranded DNA molecules which have been produced. This is the heavy nitrogen, this is the hybrid, this is the light, and this is the hybrid. Anyone has any doubt, any query, you just ask me, because I'm going to give you questions out of this particular topic. I'm going to give you questions out of this particular topic. Anyone has any query? Please ask. Okay. So see, the first and foremost thing, whatever is the nature of double-stranded DNA molecule. Now, supposingly, if initially I'm going to take N14, N14 type, and then grow it for 10 generations on heavy nitrogen, NH4Cl, and that to 15. How many number of DNA molecules will be produced? Single double stranded DNA molecule taken, grown for 10 generation, 10 cycles, 10 generation, 10 generations. How many molecules will be produced? Okay, one will produce two in one generation. In another generation, it will produce four. So how many will be produced? Can you just tell me? It would be equivalent to 2 raised to the power 10. This would be equivalent to 1024. I've just multiplied it out to calculate the value 1024. Now, out of these 1024, which would be the different type of DNA molecule? Remember, the DNA has been grown in heavy nitrogen. How many different DNA molecules will be there? See, because there were 2N14 nitrogen, and now all the reactions are being done in N15 nitrogen. So the two strands will remain N14 and N14 type. So let this be N14 type. Let this be N14 type. Others will always remain N15 types. Okay. So I'm labeling N15 as red. Okay. So the other strand will be N15 type. This strand will be N15 type, while all other strands will be N15 type. That means out of 1024, 1022 will be N15 type. N15, N15. There would be two DNA molecules of N14 and 15 type. N14, N15, and N14 and N15. केवल दो ही डीएनए मॉलिक्यूल ऐसे होंगे जो हाइब्रिड डीएनए मॉलिक्यूल होंगे, जो N14, N15 टाइप होंगे, and others will be N15 and 15 types. Whereas, how many will be N14, N14 type? ये कितने होंगे? Can you just tell me how many will be N14, N14 type? Zero. Zero will be N14, N14 type. कोई भी N14, N14 type नहीं होगा, क्योंकि N14 type के सामने क्या आ जाएगा? हमेशा N15 आएगा. There would be zero, that would be N14 and N14 type. Any doubt, any problem? Any doubt, any problem if you have? Hello? Anyone has any doubt? Anyone has any problem? You just ask me. किसी को कोई डाउट, किसी को कोई प्रॉब्लम, तो आप पूछ सकते हैं. No? Okay. So then we are going to start about the origin of replication and the process of replication. But before that, I am giving you 4 to 5 questions of this type in your group. You will have to solve all those questions. If you feel any problem, I will answer those questions. Okay. Okay. So that's all for today. We'll meet tomorrow with DNA replication and we'll start the process of DNA replication. I'm going to share this PowerPoint animation with you all. That's all for today. We'll meet tomorrow at same time. Good night. Bye bye.